I'm Leonard Zon. I'm director of the stem cell program, and my research focuses on blood diseases, particularly using the zebrafish as a model system. People often ask why I am doing what I'm doing, and when I was an undergraduate, I went to Roswell Park Memorial Institute for a summer of research, and there I worked on the leukemia ward, and I really started to understand blood diseases. So it was seemingly as if I was going to be a hematologist from the beginning. Once I started my own lab, we started studying stem cells and how clones of stem cells become transformed, and this led me to RUNX1. With regard to RUNX1 FPD, my laboratory has made a zebrafish model. And so we inactivated the RUNX1 gene in the zebrafish system, and we were able to see that the animals developed first without any stem cells at all. And then their stem cells started to recover magically. And what this indicated is there's a bottleneck during development, that certain stem cell clones are not actually able to be made. And I think this has a great impact on what the disease is doing later in life. My team right now is trying to answer questions about clonality. So what is clonality? Well, we've developed a system to barcode every cell of the body of a zebrafish. And just like when you go to the supermarket and you put things through the barcode reader, you'll know what the cell of origin is, you'll know where it came from, and when it starts to transform. And so by applying the technologies we have in the zebrafish, we've been able to find that there's a bottleneck of stem cells during embryogenesis, and that the stem cells recover over time. But those stem cells are few in number in terms of clones. This is ultimately what causes the disorder later on, both the platelet problem as well as the stem cell problem. Within the RUNX1 field, I feel there's a number of new avenues that are being pursued, which are very exciting. I know there's studies using CRISPR-based screens to find factors that will prevent the RUNX1 effect. And I think that that's going to translate ultimately to therapies. And I'm very excited about the natural history study to be able to find out how patients do over their lifetime, and this is going to instruct how clinical trials will be done in the future. I think there's a number of reasons for people to donate to societies or foundations such as the RUNX1 Foundation, and I think that these are a group of patients who really have a devastating disease at the end, and we need to figure out how to find the cures for these patients. I really feel that the investment in research ends up beginning a cycle where people in medicine are trying to find cures for patients. This is very difficult. It takes a long time to be able to do that, and research is the fundamental mechanism by which this occurs. So not only does it help the patients, it helps society, it helps financially in society, and so an investment in research at this point makes the most sense to be able to help the world and help people with very specific diseases like RUNX1 FPD actually find cures. Once I had a patient, and the patient had chronic myelogenous leukemia. And we put the patient through 2 transplants. And ultimately, the patient did well for a long period of time. But while I was seeing him, I said, someday there'll be a pill for this. And so my view is that someday there'll be a pill for RUNX1 FPD, and we just need to find out what that pill is.